Well, hello guys. We are moving on from eukaryotic cells, their organelles, and so on, to viruses and bacteria. Excuse me. These are very fascinating These are fascinating groups of cells or organisms, and they cause a lot of damage to organisms and plants or animals. And they're also very important in so many other ways that is beneficial. So we will initially just look at some diseases, okay? Some diseases, and these are not necessarily viruses and bacteria, but they're important. So basically, we have two types of diseases or conditions. We have those through an infectious agent, like a pathogen or a protozoan, right? Examples are anthrax, swine flu, herpes, common cold, malaria, salmonella. So those are transmitted from one person to another. So we say that it is an infectious disease. The others that are not infectious, in other words, if one person gets it and the person coughs or sitting close to you or hold your hand or something, depending on the disease, you are not likely to get it. And those are cancer, lupus, arthritis, allergies, hypertension, diabetes, and so on, right? So this you need to focus on the first type, the disease, its agent, treatment, and history. And we'll cover three main areas of pathology: viruses, bacteria, and parasites. Yes. So viruses, is virology, the study of viruses, bacteria, bacteriology, parasitology. All right. So we are still navigating through diseases and the surrounding issues around them. So when it comes to infectious diseases or communicable diseases, then there should be a host as an organism which provides nutrients, etc., to another organism. Then we have the parasite as an organism which lives at the expense of its host, benefits from the host. Then we have the disease, an upset in the homeostasis of the host, resulting in generation of observable changes. Then we also look at symptoms. That's the evidence of damage to the host. What shows that the pathogens are having their way with the host. It manifests through symptoms. Then we have another definition: infectious disease, one in which detrimental changes in health of the host occur as a result of damage caused by the parasite and can be transmitted. Then we have virulence, a measure of pathogenicity, which is the ability to cause disease. You have epidemic, when a disease affects a community. So at the local level, when you have some outbreak of you have a spread of certain diseases within the community, and it's more localized, more confined. It's not it doesn't spread so wide. Say it's an epidemic. Then we have pandemic, when the disease affects the world. So COVID-19, we said it was a pandemic because everyone, every continent was sort of affected. You have some more terms, disease categories. You have food and waterborne disease. You have bloodborne. You have sexually transmitted. You have zoonotic, that's carried by animals. You can be transmitted, excuse me, from one animal to another. Then we have airborne. Okay. So now we zero in on the virus. So we said that viruses are non-living infectious agents composed of nucleic acid enclosed in the protein coats and are smaller than the smallest bacteria, right? They are really, really, really very small organisms. And as you have learned in microscopy, this is where electron microscope comes very handy, so that we can get a very look at them. All right. Most biologists consider viruses to be non-living because they don't exhibit all the criteria for life. They don't carry out respiration, they don't grow or develop. All viruses can do is to replicate, and they can't even do that without the help of living cells. The cell in which a virus replicates is called the host cell. How are viruses named? Viruses such as rabies viruses and polioviruses were named after the diseases they cause, right? So after the diseases that they cause. Other viruses were named for the organ or tissue they infect. Adenoid virus affects the adenoids, causing common cold. Today, most viruses are given a genus name ending the word virus and a species name. Sometimes code names are used to distinguish between similar viruses or the same host cell. A virus that infects a bacterium is called a bacteriophage, or phage for short. So bacteriophage is one virus that we are going to use it to actually understand how viruses, attack, living cells or infect living cells, and then uses the host's internal mechanism, the cells, organelles, and the structures within the cell to actually propagate itself, reproduce itself. Okay. So one we are going to look at is bacteriophage. Remember that it has got nothing to do with bacteria, but a virus. That's a virus that infects a bacterium. Okay. So we will get closer to bacteriophage later. I want to look at the general structure of the virus or viruses, right? A virus has an inner core of nucleic acid, either RNA or DNA, right? But not both, and an outer protein coat called a capsid. Some relatively large viruses, such as human flu viruses, may have an additional layer called an envelope surrounding their capsids. So envelopes are not present in all viruses. So envelopes are made from the same type of materials or the cell membrane, right? Remember the cell membrane, what are the materials that form the cell membrane? You can put it in the comments, right? Okay. The viral structure. So viral nucleic acid is either DNA or RNA and contains instructions for making copies of the virus. That is so, responsibility of the virus. It's as if the whole existence of the virus is just to replicate itself, make copies of itself, and therefore can be highly can have a high level of virulence, or have high probability of causing disease in the organism, but it can reproduce so much of itself within a very short time. Some viruses have only 4 genes, while others have hundreds. The arrangement of proteins in a capsid of virus determines the virus's shape as well as the type of the host cell it can infect. Virus shape and size. There are so many shapes and sizes of virus. If you look into your workbook or the biology notes, I've put in there so many shapes of bacteria. Some of them are polyhedral, helical, and their size range from 20 nanometers to 250 nanometers. Come on, these are really, really small organisms. Really, really small. We are talking about 20 times 10 to the power minus 9. So if you had to write it, that would be like 0.00000000090 zeros. Then we have 2 zeros coming. You can imagine that. So we really cannot see these organisms. There's no way you can. There's no way you can see. The resolution of the eyes will not allow you to, see them. All right, so we've talked about virology and then virologists already, so let's move ahead. Now let's look at replication. There are some nice diagrams ahead that we'll look at when it comes to replication. That's one of it. That's a Lytic cycle. The other one is Lysogenic. Okay, so to get interesting as we go ahead, so stick and stay, right? So yeah, where were we? Right here. Okay, so before a virus can replicate, it must enter a host cell. The virus recognizes and attaches to a host cell when one of its proteins interlocks with a molecular shape that is the receptor site on the host cell's plasma membrane. In some viruses, the attachment protein is in the capsid or in the envelope. In the T4 bacteriophage, a protein in the tail fibers recognizes and attaches the T4 to its bacterial host cell. So you can see the bacteriophage. So you have the true structure, if you like, of the virus here. You can see the true structure of it right here. So this is more or less, the main structure of the virus, right? As you have said, having the nucleic acid and then the capsid, right? But then interestingly, has this shape, the tail and some tail fibers, very strange indeed. And this is what you say it uses to firmly attach itself to a bacteria, and then it can release its nucleic acid within it. All right, so let's get along. So viral replication. Each virus has a specifically shaped attachment protein. Therefore, each virus can usually attach to only a few kinds of cells. In general, viruses are species specific and some also are cell type specific. For example, polioviruses normally infect only intestinal and nerve cells, right? The species-specific characteristics of viruses is significant for controlling the spread of viral diseases. Okay, well, and in this way, it enables, pharmacists or chemists or whatever, producing drugs or whatever to counteract the viruses, to also know what medicines or drugs to make, knowing the types of the parts of the organism that it affects, like in the polio virus, it's in the nerve cell and all that. Okay. Viral replication cycles. Once attached to the plasma membrane of the host cell, excuse me, the virus enters the cell and takes over its metabolism. Only then can the virus replicate. Viruses have two ways of getting into host cells. The virus may inject its nucleic acid into the host cell, like a syringe injects a vaccine into your arm, like how a syringe injects vaccine to the arm. The capsid of the virus stays attached to the outside of the host cell. The second way, the envelope virus enters after attachment. The plasma membrane of the host cell surrounds the virus and produces a virus-filled vacuole inside the host cell's cytoplasm. Then, the virus bursts out of the vacuole and releases its nucleic acid into the cell. Good, so we are finally here. The Lytic cycle steps. So there are two main stages when you are looking at the bacteriophage. You have the Lytic cycle and then the Lysogenic. So we are going to look at the Lytic. Lytic, remember lysosomes, lysis, to break down, to rupture, that kind of thing. So we are going to have the virus actually rupturing a host cell, in this case, bacterial cell. And then that's after it has reproduced itself and can go ahead to infect another cell. Let's look at the steps involved. So the virus attaches to the host cell. That's what we call the attachment. B, the virus injects its nucleic acid into the host cell. That's called the entry. The host cell uses its own supplies to make copies of the viral nucleic acid and proteins. That's the replication. New viruses are assembled and ready to be released. That's called assembly. The host, then we have the host cell breaks open and releases the new viruses to continue the cycle of infection. Okay, so that is what the cycle is about. So eventually there's a rupturing of the cell, the host cell. The new viruses can then infect and kill the host cell. This process is called the Lytic cycle. Typical Lytic viruses take about 30 minutes to complete the cycle and releases 200 new viruses. You can imagine 30 minutes, every 30 minutes you have 200. Wow, those 200 then go ahead and go and infect other cells, other 200 host cells, and they will also release in 30 minutes and so on and so forth, right? So within a short time, this virus takes over completely the organism or the bacterium and then just destroys it, kills it or whatever. And in cases like Ebola and the Marburg diseases and so on, it's associated with people bleeding from their nostrils and so on. The level of pathogenicity is so high, it's so high, and within a short time, if remedy doesn't come, the person will die, bleed from the eyes and ears and nose and all that, and that will be the end of the host. Viruses are covalent because they cause disease immediately. Okay, so this is the cycle. It's a beautiful diagram of the cycle. We have the attachment, we have B entry, we have C replication, we have D assembly. After it has made so many parts of it, so it assembles it, then together it forms a virus. And you see the bacteriophage. Okay, so then from assembly, then you have lysis, then they come out and they can attack other host cells. Okay. All right, now so on to the lysogenic cycle. Well, not all viruses kill the cells they infect. Some viruses go through a lysogenic cycle, a replication cycle, in which the virus's nucleic acid is integrated into the whole cell's chromosome or DNA. Right. A lysogenic cycle begins the same way as the lytic cycle. The virus must attach to the host cell and then inject its nucleic acid. So it goes through that period of attachment and entry, right, pertaining to that virus anyway. Viral DNA that is integrated into the host cell's chromosomes is called a provirus. So a provirus may not affect the functioning of its host cell, which continues to carry out its own metabolic activity. However, every time the host cell reproduces, the provirus is replicated along with the host cell's chromosome. Therefore, every cell that originates from an infected host cell has a copy of the provirus. Right? Okay. The lysogenic phase can continue for many years. However, at any time, the provirus can be activated and enter a lytic cycle. Lysogenic viruses usually wait for an opportune time, a time when the body is not well rested, stressed, or fatigued. You can guess, some of these situations in the body, right? You have some conditions, some disease or something. So all these can lead to the immune system goes down. Then they will kick in and then begin to go through the lytic cycle. So they move from the lysogenic to the lytic cycle. Until that time, they just remain, if you like, dormant as part of the nucleic acid or the chromosome of the host cell. Interesting, isn't it? This type of virus is called temperate because it does not kill or make sick immediately, right? So you can underline the word what? Immediately. Because do not forget, it is the same virus that can actually kill. But at this time or this phase of it, it is not, causing any harm, right? It just stay within the cell, the host cell, and lies and wait. All right, we move on. Right, so we've already looked at the lytic cycle, okay, which in summary is below. Now we have the lysogenic, which is on top, right? Okay, let me take off this mark from there. All right, okay, so look at it. So we have the attachment and entry again. Okay, so bacterial host chromosome, a lysogenic virus inject its nucleic acid into a bacterium. See the bacteriophage right here, okay, sitting on top of the bacterium or any host cell for that matter, and then integrate with the host DNA or chromosome. So the viral nucleic acid is called a provirus when it becomes parts of the host chromosome. Then during any cell division, as part of the host chromosome DNA is the provirus. Right. I wonder which one is more dangerous, the lytic or lysogenic. At least we know that the initial stages is the lytic, but can lysogenic be just as dangerous? Why don't you debate about that? Okay. The provirus leaves the chromosome at this stage. So at this time, you have the provirus leaves the chromosome. So anytime the host system, you are stressed, fatigued, tired, and all that, the immune system is likely to go down, and these viruses can go through the lytic cycle and therefore cause a lot of, problems. To replicate itself, just like we saw in the lytic, and then when it is well assembled, it comes out of the bacterial cell. Then it can go on to attack other cells. All right, so we shall end on this note. We will later look at some of the diseases caused by some of the viruses. Okay, so this is where we'll end.